Interphase is the period when a cell spends 90% of its time on and normally lives. It consists of G1, S, and G2 subphases. G1, also called first gap, is when a cell grows its organelles and cytoplasm after cell division. Its genetic content is chromatin. S subphase is when DNA is duplicated in a cell. Genetic content is chromatin as well. And G2 subphase is when a cell prepares for division and organelles are in place. The genetic content is still chromatin. As the cell enters prophase, chromosomes are condensed by coiling around histones, making chromosomes visible. Nuclear envelopes start dissolving as well. Also, centrosomes start to move toward poles, and mitotic spindle begins to form as microtubules rapidly grow out from centrosomes. Each duplicated chromosomes consists of two sister chromatids connected at the centrosome, a centromere, at this time. During metaphase, chromosomes line up in the middle of the cell on the metaphase plate. Centrioles are at the poles, pulling the spindle fibers that are attached to the kinetochores on the centromere of the centrosome. The centrosomes of all chromosomes are lined up on the metaphase plate as well. More importantly, the chromosomes are lined up in a single file, which is very key to even out each side. The third stage is anaphase. In this phase, centromeres separate, parting the sister chromatids. Cells elongate, and daughter chromosomes move toward poles by spindle. The spindle microtubules that are not attached to the chromosomes lengthen, resulting in poles moving farther apart. At the end of the anaphase, two ends of the cell have equal collections of chromosomes. The final phase is telophase. During this phase, nuclear envelope starts to reform. Cell becomes elongated and the middle is pinched in. Spindles start to disappear and cytoplasm separates by pinching off cell membrane. Nuclei form and chromosomes unravel into chromatins. And that is the end of mitosis.
We have decided to exclude meiosis II because meiosis II is essentially the same as mitosis. The important difference is that meiosis II starts with a haploid cell. The first stage of meiosis I is prophase I. In this phase, homologous chromosomes come together as pairs in a process called synapsis. During synapsis, chromatids of homologous chromosomes exchange segments in a process called crossing over. Because the versions of some genes on a chromosome differ from those on its homologue, crossing over rearranges genetic information. In addition, the nuclear envelope breaks down and starts to dissolve. The second stage of meiosis I is metaphase I. In metaphase I, the nuclear envelope is fully dissolved. Pairs of homologous chromosomes move to the equator of the cell also. In addition, centrosomes are located at the poles. With this arrangement, the homologous chromosomes are poised to move toward opposite poles of the cell. Notice, the spindle microtubules attached to one of the homologous chromosomes come from one pole of the cell, and the microtubules attached to the other homologous chromosome come from the opposite pole. The third stage of meiosis I is anaphase I. Anaphase I consists of the homologous chromosomes moving to the opposite poles of the cell through a process of separation because of the spindles pulling the chromosomes to the poles. In addition, the sister chromatids remain attached, and the homologous chromosomes are the ones separating. In addition, the cell begins to elongate as meiosis I approaches telophase I. The final stage of meiosis I is telophase I, stage 4. The end result in telophase I is homologous pairs separating. Chromosomes then gather at their respective pole at each end of the cell. In addition, cytoplasm and the cell membrane begin to divide. No chromosome duplication occurs between telophase I and the onset of meiosis II. This is the end of meiosis I. Written in 1490, Leonardo da Vinci drew the Vitruvian Man. a picture depicting the proportions of the human body according to Vitruvius, an architect living during Da Vinci's life. The Vitruvian man described the human figure as being the principal source of proportion of processes in the body of men. Meiosis and mitosis are two necessary processes of an individual in order to maintain proportion in a stable and healthy body. Mitosis, the first main process in the human body. Interphase, prophase, metaphase, anaphase, and then telophase. Meiosis, the second main process in the human body that acts on sex cells. Prophase 1, metaphase 1, anaphase 1, and telophase 1.